So yeah, my talk is on the fusion of fMRI and MEG in order to obtain some spatial temporal view of the processing in the human brain. So I'm focused on the multiple domain network, and this network is a set of brain areas in the parietal, frontal, and medial frontal cortices, which seem to activate at the same time in fMRI. And this is involved in many different types of tasks, such as word, non-word, contrast, mathematical operations, spatial working memory, and verbal spatial working memory, etc. And it has been shown in previous research that the involvement of the multiple domain network is higher when coding stimulus information, when the stimulus information is harder to perceive rather than compared to when it is easier to perceive. So it seems to be a kind of flexible network which involves in processing of information with more pronounced effects in when the task is harder. But the point is that the temporal dynamics of information processing in the MD network is not really clear. And it is mainly because this network is recognizing the fMRI, which doesn't give us good temporal resolution. So in this research, I asked whether we could understand the temporal dynamics of information coding in the MD network, and whether we could utilize the spatiotemporal specificity of fMRI-MEG fusion to quantify any potential information exchange between the MD areas. So to answer the first question, we designed a task in which subjects had different stimuli, as you can see on the top of the screen, and they had a fixation which told them which button to press. So they had one set of mappings between the top stimuli and the buttons that they had to press. And they had actually two different mappings, which we called rule 1 and rule 2. And based on the color of the fixation, they had to decide which button to press. So the stimuli represented 500 milliseconds, and the subject had to press one of the buttons to indicate whether the correct, which finger was the correct response. And we actually decoded different types of information from the test that you can see here. And the coarse stimulus information is the information about whether the stimulus is on the right or versus on the left of the screen. So collapsing across any other things. So the two left stimuli versus the two right stimuli, which we called coarse stimulus information. We had fine stimulus information, which refers to inner versus outer stimuli. So the two inner stimuli versus the two outer stimuli. And we had rule information, as you can see, the two rules, we tried to discriminate the two using decoding. And also we tried to discriminate response information by decoding inner versus outer fingers, which also collapsed across any other types of information. So what we saw in the fMRI alone was that the information about coarse stimulus information was higher in the visual areas as we expected, because it's kind of periphery stimulus information, which we expected to see in the visual areas. But interestingly, what you can see here is that the fine stimulus information, which is harder, I mean discriminating between the inner versus outer, or whether the stimulus is the inner one or the outer one, which is the more difficult one, is more pronounced in the MD areas, which, as I said, was involved in the processing of more difficult information. And also we can see the rule information in the MD areas more pronounced than in the visual areas, and also the response information in the IPS, which is also associated with processing response in the human brain. And so this was for the fMRI. And what we saw for the MEG was that when we decoded the different types of information that I talked about across the time course of the trial, as you can see here, this is the time of the stimulus onset. And this shows the data on the stimulus aligned. And this one shows the response aligned data. And as you can see, the coarse stimulus information is much higher after the stimulus onset compared to the fine stimulus information. And interestingly, right before the response, the fine stimulus information, which is the more difficult aspect of information, dominates the coarse stimulus information right before the response, and it is significant. And which seems to be somehow repeating what we had in fMRI in the multiple demand network. But the point is that based on the MEG alone or the fMRI alone, or even putting them together and just interpreting them, we cannot answer the question whether they really correspond to one another, whether the domination of the fine stimulus information to coarse stimulus information before the response is attributed to the MD areas or not. So, and also about the rule and response information, we saw that most dominant information was reflected aligned to the response, which seems reasonable because the rule information is something that the subject needed to work on and retrieve, and the response information was what they responded to really. So based on the two modalities alone, we cannot decide whether actually there is any kind of connection between the MEG and fMRI. But in order to answer this question and solve the problem, we try to fuse the two modalities. And for fusing the two modalities, what we need to do and what is usually done is that people just try to come up with a kind of abstract representational space in which they can then correlate between fMRI ROIs and regions of interest and MEG to see when and where they correlate, so that they can understand when and where things are happening in the brain. So in order to do that, what we did was to obtain information from across all MEG sensors and also from voxels in specific ROIs. And as you can see here, we can have a trial from the right stimulus in rule 1, which you can see here from different voxels. You can have different activations across different voxels, as indicated by the colors here. And from the second trial, the same. You can have different activations across voxels. And then from the left stimulus in rule 2, for example, and from the left stimulus in rule 1 again, and from all the other trials, you can have representations of this different stimuli in different conditions. And what you need to do to generate that abstract representational space is to calculate the dissimilarity between the conditions that you are interested in. And what you need to do is to form a representational dissimilarity matrix, as I have here. So, for example, for this one, which is a very limited kind of representational similarity matrix, I'm showing here, you can see that the right stimulus here in rule 1 and the right stimulus here in rule 2 and left stimulus in rule 2 and rule 1, I'm just trying to see how dissimilar each condition is, collapsing across all the trials in the same condition. And what you can see here is that, for example, you can get a kind of representation like this with the right stimulus not being very dissimilar from the same stimulus in the second rule in the visual area. But it might be very different from the left stimulus because they are on the two sides of the screen, which the visual cortex is sensitive to. So, and you might see something like this with higher dissimilarity between the left stimulus and the right stimulus. So, in general, what you come up with is a kind of pattern in the representational dissimilarity matrix that you can then obtain it from different brain regions, so that you can have an abstract representation of stimulus information. And the same thing can happen for MEG at every time point in the trial. And then what you need to do is to just find a correlation between the two matrices to see when and where things correlate between the two modalities. And in order to constrain the information in the correlation that you are trying to evaluate, you add an ideal model in which you try to avoid any other interaction between different aspects of information which might intervene with the correlation which you are not interested in actually. So you add the ideal correlation. In other words, what you have when fusing directly fMRI from ROI and MEG at time point t is that what you get is this information, correlation of information. And then what you need to do is to add a model and again, actually calculate the correlation, but this time you partial out the information that is attributed to the model what you are end up to have what you end up to have is the yellow region that I show here when you parcel out the model from the correlation. And then what you need to do is just to find the difference between when parceling out and when not parceling out the model from the correlation, which is the gray areas I'm showing here. And what you will have we call commonality index between the two modalities which is constrained by the model. And you can do it by doing like repeating it across different ROIs and time points from MEG and ROIs from fMRI, and you can have different models to constrain the correlation about different aspects of the stimuli. And what you should expect after this, after calculating this commonality index, is a kind of pattern like this. Aligned to the stimulus onset, you can have like you can see where, at what time point the two actually modalities correlate. So this is a kind of commonality index you have on the vertical axis, showing that like this, the information between the two modalities peaks earlier in the V1 area, and then picks at IT area, which seems to suggest that the information is moved from the first area to the second, but we are not sure really here. So you generate different RDMs for different types of information that you might have. For example, for a coarse stimulus, as I explained, you can discriminate the right stimulus from the left stimulus. Or for the fine stimulus information, you can have inner versus outer stimuli in your RDMs. Or for the rule information, the two rules can be discriminated from one another. Or for the response, inner versus outer fingers. So what we saw in our results was that there seems to be a kind of like going from the back of the head to the front parts of the MD network. Frontal parts of the MD network, you can see there seems to be a kind of gradual shift from the visual areas. If you compare their peaks here, for example, aligned to the stimulus onset about this coarse stimulus information. And it is somehow repeated for the fine stimulus information going from the posterior to anterior brain areas. And interestingly, for the rule information also, we can see that the information about rules peak earlier in the posterior brain areas and compared to anterior brain areas relative to the time of the response where it is more relevant to happen. And the response information also is more dominant in the IPS, and it is maximum, and it happens at the latest time point at around the response time. And but the point is that like what we understand from these results is that there might be a kind of shift of information from one area to another. But what we are not sure about yet is whether there is an information flow from one area to another, which we need to quantify here. So in order to do that, based on the recent shift towards like a multivariate or like informational connectivity across brain areas, which gives you more sensitivity to target a specific aspect of information rather than like a shift of correlation between signals. We try to use this commonality index, which seems to show a kind of shift of information from one area to another, but we are not sure about that. So we are testing it here using the concept of Granger causality. So the Granger causality concept here allows you to like compare information flow from any source area to the destination area in a matrix that we will come up with. And the idea of the Granger causality is that when you you say that signal X is causing signal Y, if the past of signal X helps to predict the future of signal Y, more like up and above what you can predict from the past of signal Y itself. So the concept works like this. And so the results about this part showed that from the source area to the destination area, as you can see here, these matrices show the significance of the information flow from one area to another, with the red boxes indicating the significant results. So as you can see, none of the information, aspects of information showed above chance connectivity and flow of information from source to destination, except for the fine stimulus information, which was the more difficult information and what we expect to be more dominant, more pronouncedly processed in the MD network, which is involved in the processing of more difficult information. So what this result gives us is that we can see the flow of information from like visual areas to the IPS, also from IPS to the AIFO, and also directly from visual areas to the AIFO, and some circulation of the information within the MD network for the actually for the finest stimulus information, but not the other aspects of information. None of them reach the significance after correcting for multiple comparisons, which seem, relevant to see the finest stimulus information being circulated in the MD network. So based on the results, what we understood from like this method is that MEG fMRI fusion gives you insights about both when and where information is processed in the brain. Please note that when you have fMRI and MEG, you can make conclusions about where and when, respectively, but not when and where. So we saw that there's a sequential posterior to anterior appearance of coarse and fine stimulus information from sensory to MD areas, and the rule information seem to show the same posterior to anterior pattern. And also we saw the response information peaked in IPS before the response as a result of fusing the two modalities. And the novel informational connectivity method that we introduce here allows you to take another step and see whether there's a potential flow of information across areas, which we can determine using Granger Causality that we introduce here. And we should note that there are limitations to the fusion, and one of them is that it does not provide any additional data which is not already available in MEG or fMRI. So we should note that there is no data being produced out of the fusion. And it may also miss the meaningful patterns of information which might appear on one modality, for example, MEG, but not the other, like fMRI. So you will miss those like uncorrelated patterns across the two modalities. And it usually needs actually more signal to noise ratio, because you're like trying to fuse two noisy modalities, and what you are left with is a very noisy signal that you are working on. So you should, if we can have like higher, more like precise recording methods or facility, that would be great to do it. And I would like to finally thank you for listening and thank my mentors and collaborators on this project. Yeah, that is